Welcome to another episode from the Veterinary Research Channel. In this episode, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024, which investigates the impact of a substance called Pegbovigrastim on the immune system of young calves. The researchers, Katarzyna Dudek, Yolina Sakawa, and Dariusz Bednarek, from the National Veterinary Research Institute in Poland, explored how this compound affects the ability of calves to defend themselves against diseases in their early stages of life. The early rearing period is a critical time for calves, as they are particularly vulnerable to various infections. Often, when these diseases occur, it necessitates the use of antimicrobial drugs. However, there is a growing global effort to reduce the use of antimicrobials in animals, making it essential to find alternative strategies for disease prevention. Pegbovigrastim is already known for its role as an immunoregulatory agent in adult cows and heifers. Specifically, it is a modified form of bovine granulocyte colony stimulating factor, or BGCSF, which is primarily used to prevent or treat mastitis, an inflammation of the mammary gland, in dairy cows. Granulocyte colony stimulating factor is a type of cytokine, a signaling molecule, that plays a crucial role in hematopoiesis, the production of blood cells. More specifically, it regulates granulopoiesis, the development of granulocytes, a type of white blood cell, by stimulating the growth and maturation of their precursor cells in the bone marrow. Studies in adult cattle have demonstrated that Pegbovigrastim can increase the total number of leukocytes, which are the body's immune cells, as well as the numbers of different types of these cells. Furthermore, it is known that granulocyte colony stimulating factor can influence certain functions of human neutrophils, a specific type of granulocyte, including their ability to fight bacteria in laboratory settings. A previous study even showed that recombinant bovine granulocyte colony stimulating factor could alter the expression of certain molecules on the surface of activated bovine neutrophils in vitro, meaning outside of a living organism. Despite these known effects in adult cattle, there was limited information available regarding how Pegbovigrastim might affect calves. Only a few studies had been conducted in this younger population. One such study indicated that administering Pegbovigrastim to newborn calves led to significant increases in the number of circulating leukocytes and some specific subtypes of white blood cells. Additionally, a general stimulation of the leukocyte response was observed in calves aged between 30 and 60 days when they were exposed to induced endotoxemia, a condition mimicking a bacterial infection. The researchers of this current study had previously conducted preliminary work that suggested Pegbovigrastim administration to calves had a general stimulating effect on the numbers of granulocytes and monocytes, another type of white blood cell, as well as their ability to engulf foreign particles, known as phagocytic activity, and their capacity to produce reactive oxygen species to kill pathogens, known as oxidative burst activity, even in the presence of an ongoing infection with Mycoplasma bovis, a common bacterial pathogen in cattle. However, the assessment of these immune parameters in healthy calves had primarily been short-term, or involved only a single injection of Pegbovigrastim. Therefore, this study aimed to investigate whether administering Pegbovigrastim twice would effectively stimulate key aspects of the nonspecific immunity in clinically healthy calves. The researchers were particularly interested in examining the numbers of total leukocytes and their different subtypes, the percentage of cells expressing a specific surface marker called CD11b, the phagocytic and oxidative burst activities of granulocytes and monocytes, and the concentrations of an enzyme called myeloperoxidase, or MPO, and selected cytokines in these healthy calves. The chosen administration protocol was designed to mimic what is used in adult cows around the time of giving birth. Understanding these changes is important to determine how the effects of Pegbovigrastim might interact with the differences in the developing immune systems of young calves compared to adult dairy cattle. To conduct their study, the researchers worked with 11 clinically healthy female Holstein Friesian calves, approximately 5 weeks old. After a 4-week adaptation period, the calves were randomly divided into two groups, an experimental group of 6 calves that received Pegbovigrastim, and a control group of 5 calves that received a placebo, in this case, phosphate-buffered saline. The Pegbovigrastim was administered subcutaneously at a dose of 40 micrograms per kilogram of body weight on day 0 and again on day 7. The control group received the same volume of saline at the same time points. To evaluate the nonspecific immunity of the calves, blood samples were collected at various time points, on day 0 before the first injection, on days 2 and 5 after the first injection, on day 7 just before the second injection, and then on days 2, 5, 7, and 14 after the second injection, which corresponded to days 9, 12, 14, and 21 after the initial injection. These blood samples were used to determine the total number of leukocytes and the numbers of different types of leukocytes, including granulocytes, lymphocytes, and monocytes. Flow cytometry was used to identify cells expressing the CD11b surface marker and to assess the phagocytic and oxidative burst activities of granulocytes and monocytes. Finally, enzyme-linked immunosorbent assays, or ELISAs, were used to measure the levels of myeloperoxidase and selected cytokines, including interleukin-1 beta, tumor necrosis factor alpha, interleukin-6, interleukin-1 receptor antagonist, and interleukin-10. The collected data underwent rigorous statistical analysis to determine if the observed changes between the experimental and control groups were significant. Now let's turn to the results of this important investigation. The study found that Pegbovigrastim administration had a significant impact on the number of circulating leukocytes in the calves. On days 2, 5, and 7 after the first injection of Pegbovigrastim, there were significant increases in the total number of leukocytes, as well as in the counts of granulocytes, lymphocytes, and monocytes in the experimental group compared to the control group. These increases became even more pronounced after the second administration of Pegbovigrastim. The total leukocyte and granulocyte counts remained significantly higher in the treated calves until the end of the study on day 21. Lymphocyte and monocyte counts also remained significantly elevated until day 14. Looking at the CD11b antigen, which is involved in the migration of immune cells to sites of inflammation, the researchers observed that the percentages of granulocytes and monocytes carrying this marker were generally higher in the Pegbovigrastim treated group throughout the study, with a couple of exceptions. Significant differences were noted on days 2 and 21 for granulocytes and on days 2, 5, 7, 9, and 12 for monocytes. Regarding phagocytic activity, the percentage of granulocytes and monocytes that were actively engulfing particles did not show significant differences between the two groups after either the first or the second Pegbovigrastim administration. However, when the researchers looked at the mean fluorescence intensity, which indicates the amount of engulfed material per cell, they found significant differences. On days 2 and 5 after the first Pegbovigrastim injection, the mean fluorescence intensity for granulocytes was significantly higher in the treated group. Similarly, the mean fluorescence intensity for monocytes was significantly increased in the Pegbovigrastim group on days 2, 5, and 7 after the first injection and remained significantly higher until day 12 of the study after the second injection. This indicates that while the proportion of cells engaging in phagocytosis might not have changed, the individual cells in the treated group were more efficient at engulfing material. In contrast to phagocytic activity, Pegbovigrastim administration did not have a substantial effect on the oxidative burst activity of leukocytes. No significant differences were observed in the percentages of activated cells or the mean fluorescence intensity values throughout the study, except for day 7, where the mean fluorescence intensity was significantly lower in the experimental group. The concentration of myeloperoxidase, an enzyme released by neutrophils during their antimicrobial activity, also did not show detectable changes in response to Pegbovigrastim administration. Interestingly, the control group exhibited marked increases in myeloperoxidase concentration on days 7, 14, and 21, while the levels in the treated group remained more stable. Finally, the researchers examined the concentrations of selected cytokines, which are important signaling molecules in the immune system. The first administration of Pegbovigrastim led to a detectable increase in the concentration of interleukin-1 beta until day 7, and a similar increase was observed on day 9 after the second administration, although these increases were not statistically significant. Similarly, an increase in interleukin-6 concentration was observed in the Pegbovigrastim group throughout the study compared to the control group, but this difference also did not reach statistical significance. The concentrations of tumor necrosis factor alpha and interleukin-1 receptor antagonists remained comparable between the groups throughout the study. However, on day 21, an increased concentration of interleukin-10, a cytokine with anti-inflammatory properties, was observed in the Pegbovigrastim treated group. These results provide valuable insights into how Pegbovigrastim affects the nonspecific immunity of young calves. Now let's discuss the implications of these findings. The researchers concluded that their study clearly demonstrates that Pegbovigrastim administration in clinically healthy calves effectively stimulated several key components of their nonspecific immunity. This stimulation was evident in the significant increases in total and differential leukocyte counts, as well as the enhanced phagocytic activity of both granulocytes and monocytes. Furthermore, the study revealed a modulation of the cytokine response in the treated calves. The observed increases in leukocyte counts following Pegbovigrastim administration are consistent with previous studies in both adult cows and, to some extent, in calves. The researchers noted that the stimulation of leukocyte production, particularly granulocytes, was more pronounced after the second injection, mirroring findings in adult dairy cows during the transition period around calving. The increase in lymphocytes, while these cells are not the primary target of granulocyte colony stimulating factor, could potentially be attributed to the effect of Pegbovigrastim on the pluripotent hematopoietic stem cells in the bone marrow, which give rise to all types of leukocytes, or possibly an indirect effect of low-level pro-inflammatory cytokines on leukocyte production and release. This observation aligns with another study that reported a significant increase in circulating lymphocytes after Pegbovigrastim administration in dairy cows. The current study's findings on the enhanced phagocytic activity of granulocytes and monocytes, specifically the increased ingestion capability per cell and the prolonged activity in monocytes, differ slightly from the researchers' previous work in calves challenged with Mycoplasma bovis. In that prior study, the phagocytic activity of granulocytes was less markedly increased, and that of monocytes was not affected, although the percentage of phagocytic granulocytes was significantly elevated. These discrepancies could be due to the different health status of the calves, healthy versus infected, and variations in the experimental design, such as the timing of blood sample collection. Interestingly, the lack of a significant effect of Pegbovigrastim on the percentage of phagocytic monocytes in the current study mirrors the findings of their previous research. The absence of a significant effect of Pegbovigrastim on the oxidative burst of leukocytes in this study is consistent with the researchers' previous findings in Mycoplasma bovis challenged calves regarding the percentage of cells producing reactive oxidants. However, unlike the previous study, where the enzymatic activity of these cells was enhanced, no such enhancement was observed in the healthy calves in this investigation. These differences again highlight the potential influence of health status and experimental conditions on the immune response to Pegbovigrastim. Notably, a previous study in periparturient cows also found no significant changes in the respiratory burst function of neutrophils following Pegbovigrastim treatment, despite observing some impairment in phagocytic activity around the time of calving. The more stable myeloperoxidase concentration observed in the Pegbovigrastim-treated calves compared to the increasing levels in the control group suggests a potential protective effect of Pegbovigrastim against excessive neutrophil degranulation. The researchers propose that this could indicate a beneficial effect of Pegbovigrastim on maintaining homeostatic relationships within the calf. This finding contrasts with a previous study in peripartum cows, where an initial increased release of myeloperoxidase from blood neutrophils in Pegbovigrastim-treated cows was followed by a decrease. The significant increase in the percentage of CD11b-positive granulocytes and monocytes after Pegbovigrastim administration in this study aligns with in vitro observations where bovine granulocyte colony stimulating factor upregulated the expression of this adhesion molecule on neutrophils. This increase in CD11b expression suggests a potential enhancement in the ability of these immune cells to migrate to sites of inflammation or infection. The researchers suggest that the rapid upregulation of CD11b is likely due to the recruitment of stored integrin from within the cell granules to the cell surface. The cytokine response observed in the Pegbovigrastim-treated calves showed a trend towards anti-inflammatory activity. The non-significant increases in interleukin-1 beta and interleukin-6, both pro-inflammatory cytokines, were followed by an increase in interleukin-10, an anti-inflammatory cytokine, at the end of the study. This modulation of cytokine release is partly consistent with findings in human volunteers treated with granulocyte colony stimulating factor, where an increase in the release of the anti-inflammatory cytokine interleukin-1 receptor antagonist was observed. The human study also showed increases in interleukin-6 and interleukin-10 release, suggesting that granulocyte colony stimulating factor has an immunomodulating effect, potentially shifting the cytokine balance towards anti-inflammatory responses. In conclusion, this study provides compelling evidence that Pegbovigrastim effectively stimulates nonspecific immunity in clinically healthy calves by increasing leukocyte counts, enhancing phagocytic activity, and modulating cytokine responses towards anti-inflammatory activity. The researchers suggest that these findings support the potential use of Pegbovigrastim in preventing diseases during the early rearing period by bolstering the host's immune defense mechanisms. However, they emphasize that further clinical research is necessary to determine whether the observed increases in leukocyte count and enhanced function actually translate into a reduction in the occurrence of clinical diseases. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.